All right. So with that, hello everyone. Welcome to our Knowledge Portal focus group today. For those who don't know me, I'm Julie Jurgens. I'm a representative of our Knowledge Portal network. You're probably used to seeing my colleague, Mackenzie Brandis, who unfortunately couldn't be here today. So I'll be hosting in her stead. We're going to start with a couple of quick announcements. So we have some upcoming events, including a presentation at the Intelligent Systems for Molecular Biology Conference, ISMB, happening in Liverpool, July 20th through 24th. There we're going to be presenting on the GeneSet browser, which is a novel tool that our team has developed through the Common Fund Data ecosystem. We'll be presenting this as a part of a track from NIH on generative AI, cyber infrastructure, digital twins, and quantum computing. And the session is on July 21st. So it should be a really exciting session. I actually got to meet with some of my co-panelists this past week, and I'd highly encourage you to attend. We also have some other conferences coming up. In 2025, we'll be at the American Society for Bone and Mineral Research, September 5th through 8th in Seattle, Washington. And there we're going to be presenting on our musculoskeletal knowledge portal that we've been developing out in collaboration with the International Federation of Musculoskeletal Research Societies. So if you're interested in musculoskeletal research or have thoughts on the development of that portal, we'd love to see you there. We'll also be at the American Society of Human Genetics in October 14th through 18th here in Boston. For those who know our team, you know that this is a conference that we attend every year, and we always look forward to getting together with everyone and hearing the latest and greatest of your research in the community as well. So we're going to be running a exhibition booth for our Common Metabolic Diseases Knowledge Portal and Association to Function Knowledge Portal, as well as the Common Fund Data Ecosystem Knowledge Center. We also have a few ancillary sessions and a networking reception. We're awaiting the dates on these, but if you're attending this conference, stay tuned for those because we would love to see you there. Upcoming events in our webinar series include a public webinar for the Common Fund Data Ecosystem, where we'll be presenting on AI modeling for diabetes and precision public health. This presentation will feature guest speakers from our colleagues at Bridge to AI, who are working to develop AI-ready cohorts and tools for research. So this particular talk is going to feature guest speakers from the AI Ready Initiative, which is developing a AI Ready Atlas for diabetes research, and Bridge to AI, which is focusing on using voice as a biomarker in research. So this webinar will be Friday, July 25th, 1 to 2 p.m. Eastern Time. Finally, here are some other upcoming webinars and focus groups that we have for our Knowledge Portal network. August 14th, September 11th are the next ones in the sequence. So we look forward to seeing you there. And finally, we wanted to share a survey on behalf of our colleagues at EMBL EBI. As you know, many of us have collaborated with this team for years through the GWAS catalog, and our efforts have actually synergized to create the open resources that we look forward to sharing with you at these webinars. So if you've ever used any of these resources developed by EMBL EBI, GWAS Catalog, UniProt, Ensemble, please take a couple minutes to take this survey. This will help their team to continue to sustain their funding and to also get valuable input on the development of their resource. So with that, we turn to what you all came here for today, which is our Knowledge Portal focus group. So today we're very excited to be presenting two open portals for adipose tissue biology. One has been developed by our team. This is the Mammalian Adipose Knowledge portal, and I'm going to be presenting that first. And then we have our guest speaker, Lucas Massier, who's here with us to present adipose tissue knowledge portal. And just to give you some background, our team had started developing out a portal and then came across a paper in cell metabolism from this other group, highlighting that they had recognized the community's need for an open Adipose resource in the same way that we did. So when we came across that, we thought it would be great to synergize our efforts and had begun discussions about how we can start working together to bridge the gaps and form a more global set of resources for adipose tissue research. So with that, we can go ahead and get started. So I'm going to begin by talking about the Mammalian Adipose Tissue Knowledge Portal, or MATKP, which is an open community portal for multi-species adipose biology. I don't need to convince this group of the value of studying adipose tissue, right? It's an essential hub for metabolic regulation, involved in homeostasis, and a number of metabolic processes. And dysfunction of adipose tissue can lead to multiple diseases, for instance, obesity, type 2 diabetes, and cardiovascular disease, among others. However, there are many challenges in adipose tissue research. Adipose tissue is multifocal, meaning it's found in discrete depots, both throughout various tissues and organs in the body and within adipose tissue itself. It's complex, so there are many cell types and subtypes within adipose tissue, and dissecting which of these are actually relevant to disease processes is a challenge. The cellular composition and function can also vary between and within depots and also between species. So this makes it really challenging to study. Finally, adipose tissue is dynamic, so its composition actually changes over the course of development, as well as through various processes like nutrition, exercise, and with changes in temperature. So the sum of all of these challenges means that I'm sorry, I think somebody is not on mute. Can you please mute yourselves? Okay. So the challenge of all these conditions means that identifying a sort of longitudinal view and global view of adipose tissue is a challenge, resulting in difficulty in getting all the data in one place and getting something beyond just a single snapshot of adipose tissue under one condition. So to address these challenges, our team identified these needs and has begun putting together the Mammalian Adipose Tissue Knowledge Portal, or MATKP. This is an NIDDK-funded open community platform for adipose tissue. It features multi-species harmonized data, interactive analyses, and visualizations, and has been developed by the same team that helped to bring you the Human Cell Atlas and also the Common Metabolic Disease Knowledge Portal. I encourage you to follow along with me today. This is our URL here, matkp.org. You can scan the QR code to get there. So within MATKP, you can start by navigating to our dataset browser. This contains harmonized information about adipose tissue depots, subdepots, and other information using a standardized set of ontologies across all of these data sets. We currently feature data sets from both human and mouse, and these include single cell, bulk, and pseudobulk transcriptomic data. Importantly, these data come from multiple sources across human and mouse. So these are not just our team's data, but data acquired from other sources as well. And this is only growing. To navigate to the information for a single data set and learn more about it, you can click the Explore button here, which will take you to our single cell browser. This can also be located under Resources Single Cell Browser. Here you can get a snapshot of what you can do through this browser. You can view interactive cell type clusters available through UMaps and click on any of these to color by cell type or by other features that you select through the drop-down menu here. You can also search a gene to view which cell types it's expressed in. Navigating down on the page, you can view cell proportions by various variables, in this case by cell type. And these are available through these bar graphs, and we also have expression plots available as violin plots by cell type here. Finally, at the bottom of the page, we have the top marker genes by cell type. available through these dot plots here. You can view these for all cell types or filter just to get them for a subset of cell types. In addition, you can navigate to our differential gene expression browser, which is also available through the resources drop-down menu at the top. Here, we have differential gene expression available on a per data set basis. Select a data set here, and then select a comparison that you want to make. In this case, we're looking at insulin resistance versus insulin sensitivity. Data are available as the top 20 differentially expressed genes available through a heat map shown here. In this case, color-coded by sample groups for insulin resistance and insulin sensitivity. And you can hover over each of these points to get more information about the differential expression. Over here, you can also view volcano plots for all differentially expressed genes with upregulated genes, downregulated genes, and information about which conditions you're looking at here. You can click on any of these points to see which gene it represents, or can search a gene up here and adjust the p-value thresholds here. Scrolling down further on the page, we have the top pathways represented among up and down regulated genes available through Enricher, as well as more information about the differentially expressed genes for all genes, upregulated only or downregulated only. These are available as downloadable tables as well. If you click on any given gene, you can actually get more comprehensive information about it and its expression in that data set. So here you can stratify to view data by categorical field, in this case, BMI group, or by various continuous groups like BMI shown here. And you can change what these parameters and filters are here. Importantly, we want MATKP to be a community resource where everyone is able to contribute their data. So to begin, we have developed a data registry available through uploads.matkp.org, where you will come across a form like this. You'll be asked to log in, and then you can submit your data set and metadata. This is just in its beta phase, so we're looking for an initial group of users to test uploading their data. So if you have human or mouse transcriptomic data, either bulk or single cell, we'd encourage you to submit it now. So next, on the MATKP, we have various features that are pertinent to looking at individual genes, phenotypes, and functional annotations. So these are pages similar to those that you might have seen on some of our other portals, if you've been to them. So these feature things that are probably pretty familiar, like information about overall gene function, pathways and annotations, GTEx expression profiles, and other information about the gene's involvement in human disease. However, we also have some features that are unique to MATKP, like the one that you see here. This is a plot of gene signatures for the selected gene across all the cell types and data sets that are currently available in the MATKP. So this is something that we don't have on any of our other portals, and it's a unique value out of MATKP. And over time, we envision this page to develop out to have other sort of MATKP specific gene features. So this will be a one-stop shop for information about that gene. In addition, we have our phenotype browser, which similarly allows you to search by phenotype for a subset of phenotypes that are relevant to adipose tissue. This has features like the plot that you see here, summarizing bottom line analyses of genome-wide association studies across multiple studies for that phenotype collected by the Association to Function Knowledge portal. We also have other sort of relevant scores and information that are available here. Here we have functional annotations that are available through the resources drop-down menu. And this has information collected by our colleagues at the Common Metabolic Diseases Genome Atlas, or CMDGA. This includes functional annotations, including epigenomic and transcriptomic data pertinent just to adipose tissue, and showing you various relationships for those annotations. So here you're getting a sense of the overall expression of this gene in adipose tissue, as well as how specific its expression is to adipose tissue. Finally, we're building out an educational component to the MATKP. This is symbolized through these anatomograms that are available through our landing page, as well as an About Adipose drop-down menu that we have from the top navigation bar. Importantly, this is something that we're working actively to build out. We envision this page to expand to have other information such as mini reviews on specific topics like lipolysis or thermogenesis. And we also envision including video protocols that are relevant to adipose tissue. So this is something that we're actively expanding, and you can stay tuned for those developments. So finally, the best part of the MATKP is this is just the beginning. We are a multi-year project funded over a 5-year course, and we look to expand to engage you as our community of users to submit your data and tell us what matters to you. We're also expanding the number and types of data sets that we host, species to include other species, for instance, non-human primates, and understudied adipose tissue depots. These are some of the other data types that we want to integrate over time. Multiple different omics data sets, epigenomics, lipidomics, metabolomics, multiomics, proteomics, imaging data, more genetic data, and things like cell-cell interactions. And finally, the purpose of our webinar here today is to highlight this new global partnership to make adipose tissue data more accessible on a global scale. So as I mentioned, we recently begun discussing with the team that has been developing the Adipose Tissue Knowledge Portal about how we can synergize our efforts and work together to build up these portals and make both of them more visible and used. So we're excited to feature Lucas as our next speaker to talk to us about that today. If you want to access both portals, you can go to this URL here, and that will take you to a unified landing page with a high-level overview of what our portals have to offer and links out to both. So in summary, MATKP is an integrative resource to accelerate adipose biology research. It can unlock insights into adipose tissue function and implications in metabolic disease. Engaging you as our community of users is key to our evolution. We want to make a resource that is actually useful to people and that people can feel involved in. So stay tuned for updates and developments and feel free to reach out. And with that, I would love to acknowledge our amazing team that made this happen. Everybody is highlighted here, as well as our funding through NIDDK. And with that, thank you so much. And I'm happy to take a couple of questions.
I was just wondering if you foresee that there's going to be any ability to download the data, or if it's just going to be an upload only kind of source. Because I know like right now trying to pull data from like Geo and other sources has been kind of a headache. But being able to download data from one source would be really awesome.
Yeah. That's a great question. I think we do have the ability to download some data types. I would say it largely depends on the type of data that you're looking to access. If it's anything, that has anything protected, individual level, anything like that, that is not something that we are going to be making accessible for download. But for more summary level data, we will make those available to download. Thank you. Any other questions for now? If not, we can turn to our next speaker. All right, fantastic. So, Lucas, feel free to take it away.
Okay, I'm sharing my presentation. Maybe kindly let me know if that works. So Okay, thanks for the invitation, and yeah, I'm presenting the Adipose Tissue Knowledge Portal team here to present our portal. We need to, yeah. So as Julie said, we recently published a brief description of the portal in Cell Metabolism, where you get some top level information. And here in the image, you kind of see how it looks like. And we separated our portal in 8 different modules. We have a gene summary, which kind of summarizes the most important information. And then we have a clinical module, a module on depot differences, on cell type differences. We also included some single cell and spatial data. We also included in vitro perturbation data sets, and we have a gene finder module, and then some about section and also information on how to get started, and then a main search bar that leads to the gene summary section. Okay, so just some very brief technical summary. I mean, we built the portal in Vue, and most of the interactions are built in Shiny because, I mean, we are more kind of computational scientists, or like coming from also from a wet lab field. So for us, it was important to translate what we do in a normal analysis to the portal, so that the figures are also similar to what users as us like expect, and that's why we build it in Shiny, which also allows download of, for example, ggplot objects or something like this for further refinement of the users. For the gene finder, we have an MySQL database, which we access using Flask, and all the code is available on GitHub. So if you want to contribute, if you find some errors, if you think you can do better, we would be happy to get your feedback and implement it. Okay, but now for the portal itself, our first part is about data collection and pre-processing. So our main goal with the portal maybe was also initially a bit selfish, because we do have both at Karolinska in Stockholm and also here in Germany quite large bulk transcriptome data sets with like detailed clinical data available, and we, due to ethical permits, cannot share the clinical data. So we were often contacted by other researchers that we should look at their candidate genes or whatever to confirm findings. And we're like, okay, this takes up a lot of our time, so why not make these data sets available in a way that we can share aggregate data or like correlation analysis, but not the clinical data itself. And in this sense, we collected all the clinical data sets that we have in-house, but also through collaborators or publicly available data. And in total, we collected up to 60 data sets now across these modules. And we have transcriptome data of about 6,000 individuals totally now in the Adipose Tissue Knowledge Portal. For the clinical module, you can see here that this spans quite a wide range of age, also BMI. So we have lean individuals, also people living with obesity, and we also span a broad range of HOMA values. One thing that we did is that we, for the portal, so that the data is actually comparable, we, if available, went back to the raw data and reanalyzed all the raw data in terms of adipose tissue specific transcriptome data sets, that is mostly microarray data sets, which we also remapped using both the gene code and the phantom data set to get a better coverage of long non-coding RNAs. What we observed, and what was also already known before, is that actually if we reanalyze the microarray data sets from scratch, is that the probe annotations provided by the manufacturers of these arrays are actually wrong, because they were never updated since the arrays released. So what we observed is that actually when we reanalyze everything, we identified quite a lot of probes that were either clearly wrongly labeled in the annotations, that had missing items, extra items, and partially correct items. And I just have 2 examples here. So for example, for this one probe, and the probes are always shown in orange here, the old annotation was for the gene TMF1, but if we map the probe to the genome, actually it's not even found in the TMF1 gene, but on a completely different chromosome in the POMK gene. So this was clearly wrongly labeled. We also found some partially missing items, for example, that these probes here were mapped to the HCAR3 gene, which is correct in the annotation, but we found that it's actually also mapping to the HCAR2 gene. So then the correct annotation should be both for the HCAR2 and HCAR3 gene. So we did this for all the array data sets that we had available. And so these are all the array types that we included. And you can actually see that according to our analysis, at least 25% of the probes were actually incorrect or partially incorrect. And we can improve the coverage and the correctness of the probes, both in the coding genes and especially in the non-coding genes, as a lot of these probes were designed before we actually knew much more, or like as much as we know today about long non-coding genes, like where they are and what they are called, etc. But so we think we can actually also improve older data sets, which also makes a reanalysis maybe interesting. The benefit of having multiple cohorts is, of course, that we can compare them and get more accurate results. And this is probably as expected. So here in this example, we compared people living with obesity versus living without obesity for a number of cohorts in the Adipose Tissue Knowledge Portal. So here we take any combination of two cohorts and compare them to each other and analyze the similarity of results. And you see that the more cohorts you take and compare with each other, the more similar the results get, as we would expect. And this is also highlighted if you take, for example, a single gene, again, comparing people living with obesity to without, that if we take any combination of two cohorts and compare them with each other, we have quite a wide range of effect size for this gene's idea. But the more cohorts we include in the analysis, the more we get maybe to a correct effect size of a given gene. And I think that is the strength of our portal now, that we have so many clinical cohorts from different hospitals, different array types, that we can easily generate meta-analysis to get to maybe to the correct effect size. So this is the result from all genes. This is just highlighting the fact that we have now a better coverage of non-coding RNAs compared to the old array annotations. So we find quite a number of coding genes that are regulated by obesity across 16 cohorts. I think this is now 2,500 individuals, but also quite a number of long non-coding RNAs. And we compared that. So we have about 900 non-coding RNAs that are regulated by obesity and about 750 coding genes that are regulated by obesity. And there were 108 where the non-coding RNA had transcriptional start sites within 5 kb of the coding gene. So we thought that they might be co-regulated. We also compared that to other data sets that are available in the portal. So we have also data sets on weight loss, both diet-induced and surgery-induced weight loss. So here we compared it just to the surgery-induced weight loss. But for the coding genes, we see that genes that are linked or that are upregulated in obesity are normalized with weight loss. And if we compare to the single cell data, we can also link them quite clearly to specific cell types. In contrast, for the long non-coding genes, they are not as clearly linked to the regulation after weight loss. So we have also some inverse regulations, and we cannot really yet link them to specific cell types, usually because the single cell data is not deep enough to cover the long non-coding RNA genes. We also explored a bit then the overlap between the non-coding and coding genes and looked at these 108 genes that are transcriptionally close, and we found that most of them are have the same regulation in obesity, so they co-correlated maybe, and they also correlate with each other. And we did a subsequent transcription motif enrichment analysis and also saw that in these long non-coding and coding genes that are in the same transcription region, we have some shared transcription motif patterns, which might suggest that they are co-regulated. But these are things that we think or hope that we can help identify with the data in the portal. When we did the portal, we also observed that there's barely any published protein cohort on adipose tissue. So there were two smaller cohorts with about, I think, 20 individuals. So for the portal, we made a new proteome data set. So this is 100 individuals, of which 50% are men, 50% are women, and 1/3 are lean, 1/3 are overweight, and 1/3 are obese. So we think we have a quite good representation of body weight spectrum, and we generated proteome data. From that, here again, the clinical parameters. So we also spent quite a range of age, BMI, as the groups indicate, and also insulin sensitivity. This is just a basic PCA plot showing the differences between the BMI groups. But this proteome data set is also freely available for analysis in the portal. Okay, so this is how we collected our data. And then I hope that the videos are working now. So this is the gene summary if you type in the search bar. So you get a summary of all the modules. On sex differences, clinical correlations, you have the proteome data. You see also obese versus non-obese. You have the weight loss cohorts. You see depot comparisons. We have in vitro data on adipogenesis, perturbation data, and also some basic single cell data. And you can download everything as a PDF file so that you can use it in your publications. So feel free to test out the summary also. We have then so the summary just gives top-level information, but if you want to have detailed information and toggle certain cohorts or change different parameters, you can go into the other modules. So here's an example of the clinical module where I just want to show you how you can generate analysis. So you can here toggle on or off the clinical cohorts. You can select a wide range of traits, and you can also adjust to any parameter. And then you can type in your gene. And so here, for example, it's very easy to generate meta-analysis for a given trait. And you can toggle, like if you only want to have cohorts with obese individuals or lean and obese, so you can select the cohorts you want. You can also get the data as table format, and you can download everything as PDF or as text file. We also pre-calculated a meta value across all cohorts if you want to compare across multiple. You need to select correctly. But if you want to compare across all cohorts, across multiple clinical parameters, you can use the pre-calculated meta values, and that, for example, would give you then a heat map that indicates also significance. And then, so here we also have, for example, weight loss. So you can select cohorts on bariatric surgery, but also on diet-induced weight loss. And the last example that I put here is we also have in vitro data, for example, on adipogenesis. I need to copy my genes again. So we have data on 2 different cell lines. And so here you see the time points and you see the regulation with adipogenesis. We also have protein data for some of these time points, and you can also generate heat maps for that. You can select the different times, and then we have different other data. So this, for example, is a fractionation that we did where we FACS sorted different cell types and did bulk array on that. So there's a plethora of data And we hope to extend this also, which I cannot show now everything. But these were just some examples. And for the data that don't contain clinical data, you can also download the ggplot objects. So if you want to change colors, etc., you can also directly load them into R. We have some basic customization options here. So if you want to change colors, you can go to customization. It also allows you to change the clustering method, etc. And we maybe also can extend this in the future. Okay, so then we have one very brief section on showing how you can use the portal to come also up with new candidate genes to translate into in vitro studies. So here we use the gene finder module and we select the genes that are adipocyte enriched and that correlated with basal or isoprenaline stimulated lipolysis. And so we came up with a list of genes that were overall data sets where we have the lipolysis measurements in the portal linked to lipolysis, and they're randomly selected 18 targets just for a mini screen. This is just showing that if we knock out PPAR gamma, which was in the lists, that we see an effect which is kind of a good positive control for us. And then we performed a mini screen showing that, so these are fold changes for stimulated lipolysis, and we show that indeed 5 of the selected genes had a strong significant result on stimulated lipolysis. And then we did some brief literature research and focused on Pyro3. And we went then again to the portal. So these, for example, are plots from the single cell module. We see that Pyro3 is enriched in adipocytes. And this is then a plot from the adipose tissue fractionation, where we have FAC sorted these cell types. Well, we did a collagenase digest, so the adipocytes are floating, and we did transcriptome there. And the rest of them are FAC sorted and then transcriptome done on them. And this nicely confirms that the Tyro3 is expressed in adipocytes, and its 2 ligands, PROS1 and Gas6, are mostly expressed in fibroblasts. So these are also plots taken directly from the portal. We also performed a meta-analysis for the Tyro3. We see that it's upregulated in people in lean subjects, which we also could confirm in our new protein cohort here. And so we thought, like, in this way we can show that we can use the gene finder module, we can select our criteria and come up with new genes, and then we can use the other modules to explore these genes if they are actually clinically relevant. And we did some very tiny in vitro validation. So we stimulated with the Tyro3 ligand Gas6, and we did see that it leads to both an increase in basal lipolysis, but also an increase in isoprenaline stimulated lipolysis, thereby confirming the results from our hit. Also did some preliminary Western blots showing that the treatment indeed leads to a phosphorylation of STAT and ERK. So this is just one example of how we envisioned that one could use the gene finder module to actually use the portal to find new ideas for in vitro studies. So what are our next steps? Of course, we want to improve the portal. It's currently just optimized for large screens, so it would be great to work on a mobile version. We need to improve the responsibility and the speed. Sometimes it's still a bit lacking. And we also need to improve the documentation. We, of course, plan to continue the integration of new data to existing modules. So if you have data that you think should be in the portal, please contact us. I think we also have a contact formula on the portal itself. We are also planning to develop new modules, for example, on epigenetic data on lipedema, for example, or others. Of course, we want to integrate feedback. So if you have feedback, if something is not working, please best comment on the GitHub page so we can directly address it. And most importantly, we want and need to find structures to allow the contribution of the adipose tissue community for a continued development. And this is why I think it's great that we are in contact across the adipose tissue portals so that we can like combine strengths and learn from each other and also focus on different strengths. And but like, yeah, I mean, for us, it's really we hope at least that the community uses these portals and also provides their data and helps them in continued development. And I think to generate living portals that grow and are used, I think it's necessary that the community also kind of can contribute to that. But of course, that needs some structures. And with that, I want to thank you for your attention. And yeah, I'm presenting on behalf of our portal team, which was mostly done by GRA and Danai and Michael and Niklas at KI. And we have a scientific advisory committee of Christian Wolfram, Matthias Fleur, and Dominique Lanchon. And of course, the portal was only made available through the data of many collaborators, which we also like to thank for that. Thanks.
Fantastic. Thank you, Lucas. Any questions for Lucas? See, we have a question in the chat from Doug. It seems like the two portals are complementary. Yes. Are you working on fully integrating the two of them? So if you enter one portal, you can access data and visualizations on the other. That is a great question, Doug. We do think that this is somewhere that we can go in the future. And the link that we put up in the chat above to access both the portals is sort of a preliminary step toward that. As a next step, we're working on developing API connections so that we can fully integrate. So say if you search a gene, you can get results from both portals all in one place. So that is something that we've actively been discussing and a longer term vision that we can see. Thank you.
Yeah.
Can I add one thing? So this is Michael. I think these are very different portals, but as you said, they are completely complementary and they have completely different strengths, which I think and integrating them would be a major endeavor. I don't think that's possible, but having a common entry portal and achieving, aggregated results in a simple manner in the entry page. That's something that we really look forward in developing, and we've come a long way already, I think, in this endeavor. But having them, joining them into a similar infrastructure and computational bioinformatics frame, I don't think that's possible. But again, I'm not a bioinformatician, I'm a clinician. So we might add something.
Yeah, I think we will continue our exchange and see how it's possible for certain modules, maybe for other modules not. If we develop new models like that we don't have yet, for example, on lipidema or epigenetics or whatever, I think we could talk beforehand so that we align much better how we can integrate them into the portal. So we will see. And then there was another question, are there any overlaps in the data used for both portals? I don't think there is yet much overlap in the data used. So, Julie, correct me if I'm wrong. We did use Margot's single nucleus and even state data set in our meta-analysis that we did. So in a sense, that is integrated. But I think from the clinical data sets, there's none that are shared.
I think that's right. And I think, as others have pointed out, that's kind of the beauty of these two portals, right? We have developed out things from a different perspective that's somewhat complementary. The ATKP team has integrated a lot of the clinical data, also the proteomics data that Lucas showed, and our team has integrated in multi-species data. So I think that's a nice feature about having two portals at this point.
Yeah, just to add in, I think both portals right now have a single cell data browser, and so there is some overlap in the specific single cell data, but we're both also going in kind of different directions onto the types of data that we want to add. You know, we're talking about imaging data for the MATKP, and like we discussed, the ATKP is more clinical data. So I think that we're going to continue to diverge, but hopefully still, like we said, have complementary data sets and things to learn on both portals.
I think a great thing from our side was also we were considering at an early stage to develop a mouse module, but given now this collaboration, that's completely redundant. So we've refrained from doing that, and we focus on the human aspects. And I think that MAPTK is really important in providing that other perspective on other species, where that can be developed even further with additional species as we go along.
We have another question in the chat, not quite understanding how this single cell data related to weight loss by surgery. Do you have longitudinal data?
Longitudinal bulk data. So we have two cohorts on with a 2 and a 5 year follow-up on bulk transcriptomics, and we have 4 cohorts on diet, which is usually, I think it's an 8-week or a 16-week 800 kilocalorie diet or something. We do have the baseline single nuclei data from the recent Nature paper by Ferdinand von Meyen, but we had no time yet to include the weight loss aspect of this data set in the portal, which we are planning to rework our single nuclei module to better allow group comparisons, for example, for the published weight loss single nuke data. So this is available, but at baseline, but not as a longitudinal view yet, but we do have the data and it's published also.
Great. So since this is a focus group, we also wanted to open up to questions. Do folks who are in this Adipose community, are there things that you would like to see that we have not yet developed with our respective portals? Any future directions that people can envision there? But if you think of things not on the spot, feel free to reach out. I think both of our help desk emails are right there on our site, so can feel free to chime in. Any other questions? All right. Well, if not, thank you so much. Thank you, Lucas, for joining us and presenting your wonderful resource. And thank you to the whole ATKP team for the wonderful collaboration, to the MATKP team as well for your work in developing out our portal. And we're excited to move forward with these resources. All right, thank you.
Thank you.
Bye-bye.
Bye.